Hi everyone, I'm Charlotte Kilpatrick reporting for Vaccine Nation at the World Vaccine Congress in Washington. I'm delighted that today we're joined by Dr. Bret Sellman of AstraZeneca. Thank you so much for your time.
Thank you for having me.
To kick us off, would you kindly just contextualize your role in the organization?
In AstraZeneca, I work in the vaccines and immune therapies therapeutic unit, and I head the bacterial vaccines discovery team.
Fantastic, that sounds like a very important team to be on.
Exciting.
Great to hear. So your panel at the workshop yesterday explored the importance of escape and priority pathogens in the problem of AMR. Can you tell us a little bit about the key concerns that came up during this?
One of the key concerns is that AMR, antimicrobial resistance, is really one of the panelists said this, a hidden pandemic. So we aren't really aware of it, but it's a serious problem and is a challenge to modern health as we see it. And they're all difficult pathogens when thinking of trying to prevent them with either vaccines or monoclonal antibodies. So I think we need to be very deliberate about antigen selection, how we want to target these bugs, what virulence factors we want to target. They're difficult, the pathogens that are left, so we probably want to target multiple antigens, so probably be combinations. And we also need to be very deliberate about how we design clinical trials, because even though the problem is large, the overall infection rate is low. So we really need to run studies to better understand how we identify patients at risk and enroll those patients so we can have clinical trials that will be in as shortest period of time as they can be in to be able to understand if the vaccine or monoclonal antibody is working.
Absolutely. So you've just mentioned the role of monoclonal antibodies in there. How do they fit into your team's strategy, and why do you consider them a sort of worthy pursuit, to speak?
Yeah, I mean, monoclonal antibodies at AstraZeneca are a cornerstone of our strategy. We were born out of MedImmune with a large history in monoclonal antibody discovery against viruses, but we also have a lot of expertise in antibacterial antibody discovery. In fact, I've been there 16 years, and it's where we spent the majority of our time and efforts. So we're using that experience in monoclonal antibody discovery really to launch us into the future, while at the same time informing us about what antigens and how we want to approach antibacterial vaccine development as well.
Great. So you just mentioned, I love the idea of launching into the future. We're hearing a lot about innovative technologies and developments that are happening at the moment. Are there any specific technologies or processes that you're using or seeking to sort of accelerate your process?
Yeah, absolutely. One thing in particular, we all learned about this during COVID, is mRNA technology, delivering a vaccine as mRNA. We've been partnered with DARPA for maybe 10 years, actually developing mRNA technology to deliver IgG. So we think there are a lot of advantages there, could drive down cost of goods, but also reduce the time from discovery and also for manufacturing to getting into people. So mRNA technology is one of the key things. Also, we want to leverage some of our successes in the past, discovering and developing multi-specific and bispecific antibodies against pathogens. So we can take that combination of antibodies and put them into a single molecule, which is easier to deliver and more cost effective.
Fantastic, and it's interesting to talk about delivery. We've had conversations with some of your colleagues about the importance of delivery after you figured out the development, just focusing on actually what are we going to do with it. So that's really great to hear this sort of alignment throughout the Congress.
Yeah.
My final question is, what are you looking forward to for your time here? Or when you leave, what are you hoping you will have achieved?
Really, it's really exciting to be here. To touch base and with my friends from the past. Actually saw someone yesterday who I haven't seen in over 10 years. He used to work at AstraZeneca in our former infectious disease group, and we sat down and talked for a while yesterday, so it's really nice. I also hope to make new connections, networking, collaborations, and also learn about what's cutting edge, what can help us with some of what we just talked about, make things faster, make things better.
Fantastic. Thank you so much for your time.
Thank you.